This is a chapter summary of 16, 17, 19.1, and 19.2 by Mackenzie Foy and Heather Roy. Chapter 16 is entitled The Molecular Basis of Inheritance. Nitrogen bases that go together are adenine and thymine, adenine and uracil, guanine and cytosine. Adenine and guanine are considered purines, whereas thymine and cytosine are pyrimidines, which is why they go together, because purines and pyrimidines always bond together. RNA is single-stranded, and the sugar is ribose. RNA folds into a 3D shape and contains adenine, uracil, guanine, and cytosine. Thymine is not included in RNA. DNA is double-stranded. Its sugar is deoxyribose, and the nitrogen bases in DNA are adenine, thymine, guanine, and cytosine, not uracil. Some vocabulary to know on the next slide is helicase, which unzips and breaks the hydrogen bonds, single-stranded binding protein, which helps the bubbles stay apart after parental strands separate. It binds to the unpaired DNA strands and keeps them from repairing. The replication fork is where the DNA split. DNA polymerase III adds new nucleotides, sugar, phosphate, and a base, and makes a nucleoside. Ligase is when links strands together and connects the Okazaki fragments. Lagging strands are the Okazaki fragments. They go from 5 to 3. A leading strand is a continuous strand, which is the opposite of a lagging strand and goes in the direction of 3 to 5. Topoisomerase keeps DNA linear. It helps relieve the strain in the double helix by breaking, swiveling, and rejoining DNA strands. Telomeres are a protective cap at the end of a chromosome. Telomerase rebuilds. If there isn't a telomerase, the DNA gets smaller and smaller and eventually blows up, as in apoptosis. Cancer cells are active telomerase enzymes. Next slide. Chapter 17 is entitled From Gene to Protein. The key concepts to know are that replication is going from DNA to another DNA. Transcription is going from DNA to RNA. And translation is going from RNA and making proteins. The flow of information in cells starts in the DNA, goes next to the RNA, and then to proteins. There are 3 kinds of RNA. There's messenger RNA, transfer RNA, and ribosomal RNA. Next slide. These 3 kinds of RNA more in depth are messenger RNA, which carries DNA message from nucleus to cytoplasm. This message is carried in codons, and AUG is always the beginning of the start of a codon. The stop codons are UAA, UAG, and UGA. In prokaryotes, this messenger RNA is functional as soon as it is transcribed. In eukaryotes, however, messenger RNA must be processed before use. The GTA cap is added to the 5 end for stability. Poly A tail is added to the 3 end for stability. Transfer RNA brings the amino acids to the ribosomes. It attaches amino acids in their proper place. Amino acyl tRNA synthesizes enzyme and attaches a specific amino acid using energy from ATP. Ribosomal RNA is made up of 2/3 ribosomal RNA and 1/3 proteins. The large and small subunits join to form functional ribosome only when attached to the messenger RNA. It binds messenger RNA. It has 3 binding sites. The first is A, transfer RNA with new amino acid. The second is P, where a peptide bond forms, growing a peptide chain. And the third is E, where the tRNA exit. Next slide. Transcription more in depth. It occurs in the nucleus. The initiation of transcription is when the RNA polymerase binds to DNA at a region called a promoter. It can only attach to nucleotides in a 5-3 direction. Elongation. RNA chain grows in the 5-3 direction. The non-coding strand of DNA reforms a DNA double helix by pairing with the coding strand. Termination. Transcription proceeds until RNA polymerase reaches a terminator site on the DNA and an RNA nucleotide is then released. Translation. It occurs on ribosomes in cytoplasm. The first is initiation, where small ribosomal subunits attach to the 5' end of the messenger RNA, which is the start codon. The energy for this comes from the GTP. The second step is elongation. Ribosome moves along the messenger RNA, matching the transfer RNA anticodons with messenger RNA codons. Last step is termination. This occurs when the ribosome enters a stop codon. Polysomes are strings of ribosomes that can work on the same rRNA at the same time. Next slide. Mutations. When found, they're usually not harmful. They can provide genetic variability. They are the foundation for natural selection. They can be spontaneous. They're caused by mutagens, and carcinogens are what cause cancer. This is cell mutation. When they're found harmful, they can change the protein function. A point mutation is a change in one base pair of a gene. A silent mutation is changes coding for another amino acid. And a nonsense mutation is when the code changes to a stop codon. Moving on to Chapter 19, Viruses. Viral structure is when viruses are not cells. They are considered non-living because they can't replicate their own DNA. They are infectious particles that consist of a nucleic acid and a protein coat. They have a capsid, a protein shell that encloses the viral genome. Capsosmeres are their subunits, and they can be double or single-stranded DNA and double or single-stranded RNA. Viral cycle reproduction is called lytic cycle. The steps are 1, attachment, 2, injection, 3, hydroxylation, 4, assembly, and 5, when they release. The result of a lytic cycle reproduction is the death of the host cell. Bacteria have defenses against phages, including restriction enzymes that recognize and cut up certain phage DNA. Another type of viral cycle reproduction is lysogenic cycle. It is a temperate virus, so it doesn't kill the host cell, but may give rise to the lytic cycle. Genome replicated without destroying the host cell. The genetic material of virus becomes incorporated into the host cell DNA. That's all we have for chapters 16, 17, 19.1, and 19.2.